co-authors, I want to thank you for coming to our presentation, Eco-evolutionary Feedbacks Predict the Time Course of Rapid Life History Evolution. We're part of a large and magnificent collaboration with a lot of other people, and I'll be drawing on some of their results as well as I make this presentation. Now, because my title has no real content, I thought I'd actually tell you what I'm going to do. So this is the take-home message. Eco-evolutionary feedbacks predict, as where the little pointer is, the time course of life history evolution. In order to drive that take-home message home, first I'm going to talk about the conceptual background, feedbacks, and their importance. Now, when you hear that, you think, why is this even interesting? Right? That's your job. Second thing, I'm going to talk about guppies and why guppies are a suitable system for looking at this question. And that's when you think, oh no, not another guppy time. And then finally, we get to the point, the time course of rapid life history evolution, where you think, is he getting to the point yet? So that's the roadmap of the talk. So I'll begin with the conceptual background, feedbacks, and their importance. And I think that's important because eco-evolutionary feedbacks has become almost an advertising slogan in a lot of areas. And I think it's important to say what I think they mean and why I think they're important. So I begin with this quote from Flussman et al. Evolutionary and ecological dynamics are likely to be codependent when changes in gene frequency produce changes in phenotypic traits that crucially affect interaction strengths. And by interaction strengths, they mean the per capita coefficients in which two species interact with each other. Now, this idea that there would be genetic variation in interaction strengths is not new. This dates back at least to the 50s and 60s. Up the upper left, Thomas Park and Lonnie Lloyd, tribolium strains in the 50s and 60s, and demonstrations in the 70s and 80s that natural populations harbored standing genetic variation for interaction strengths. Now, that's not really feedbacks in and of itself, it's an element of these feedbacks. The feedback idea can be traced back to David Pimentel in 1961, a little later to Dennis Chitty in 1967. And by these feedbacks, what Pimentel and others have meant is that you have this double feedback loop between genetic variation on the left-hand side and some aspect of the environment on the right-hand side, in this case, the density or phenotypic array of another species, and what happens is that this array of genotypes, through their interaction coefficients, through competition, predation, parasitism, exerts an effect on this other species, which then responds via the response to selection by creating a different array of interaction coefficients that produces a selection pressure back on the focal species. And this little loop revolves around until it reaches an equilibrium. The key being that in the focal species, they affect their environment, defined in a variety of ways, in a manner that actually changes the selection pressure on them. The resurgent interest in the modern day of this topic really is articulated well by Tom Shaner, who says that rapid evolution opens the possibility that evolution can occur so quickly that ecological and evolutionary change may be commensurate in time and in feedback loop. In other words, selection is sufficiently strong so that allele frequencies and numerical dynamics occur on a common time scale. Well, who cares? I mean, why is this interesting? Well, this is important in ecology because when that happens, the interaction coefficients change with sufficient rapidity from one generation to the next that you can take a system that perhaps began in a region of parameter space that would have made it unstable, and by changing the interaction coefficients quickly, can move it into a region of parameter space where it could become stable. In other words, they change fast relative to the population size, so that these things can stabilize certain types of ecological systems. And in the search for coexistence, this becomes very important. So ecologists find this fascinating because it allows an answer to the paradox of coexistence. It ought to be fascinating in the study of evolution because these dynamics typically are density and frequency dependent, which puts them firmly in the realm of soft selection as described by Bruce Wallace a long time ago. The interesting part about this is that this is a qualitatively different form of selection. We know that in hard selection, for example, selection maximizes mean fitness, as Ruth Shaw talked about this morning. In density-dependent selection, selection maximizes population size or the number of individuals in the susceptible age class, and in soft selection, selection maximizes nothing. What it does is optimizes some function of population size and interaction coefficients, and it can even minimize population size. So it's qualitatively different. And it is really part of the explanation for, as Brian Charlesworth tried to answer this question, why we are not dead 100 times over. So this is a very important concept. Now that's the theoretical motivation for the question. What about guppies? Applying the issue to guppies. And I say the issue to guppies and guppies to the issues. This is the part of any talk or presentation where someone says, guppies are great to study this issue, but they neglect to tell you whether the issue actually applies to guppies. Does the natural history tell us that it should be studied? Just because you can study something doesn't mean you should. I'm not kidding. But I digress. I promised myself I wouldn't. Okay, well this is the phenomenon we're trying to explain. These are an array of guppies, and this is picture courtesy of Helen Rodd. On the left-hand side, these are from what are called a high predation community. On the right-hand side, a low predation community. And I want you to see that not just the fact that they're more colorful on the right-hand side, they're larger. Because males do not grow after maturity, this means they matured later and larger. So what you're going to remember now is low predation, later, larger. Low, later, larger. Low, later, larger. If I had more time, I'd make you say it back to me a few times. But that's what we're trying to explain, why this is so. Now, this problem presents itself to us in the northern range mountains of Trinidad, where the streams range from a typical pool riffle structure at high elevation to a more modest, wider, shallower structure at low elevations. There are two major community types that we need to think about. There's actually three steady states, two that I'll talk about. This one at the low elevation is a high predation location. There are a number of piscivorous species that make a guppy's life nasty, brutish, and short. The other community is a low predation location where guppies coexist with only a killifish. And the reason they become these discrete communities is that they are separated by barrier waterfalls. The larger predaceous fish do not get above these barrier waterfalls, but guppies and killifish do. There's also a community only of killifish further up, but I'm not going to talk about that today. So this is what we have. And so you notice two things about this cartoon. One is that there are predators and fewer predators. There are fewer guppies, more guppies. Predators do two things. Directly, they increase the mortality rate of guppies. Indirectly, they increase the density of guppies, at least the absence of predation increases the density of guppies. So you have a direct effect of predation that can influence the life history, and an indirect effect. So which is it? Is it the predation effect, the direct effect, or the indirect effect of density that's selecting for the later size, later age and larger size? Well, the original hypothesis was predation. If it were predation, then we would expect mortality to look like it is on the left-hand side, or the survival rather, I'm sorry. The predicted survival should look like this pattern with the low predation of the solid lines, the high predation of the dashed lines. The data look like this. And I don't need to go over it in detail to tell you that the right-hand side does not look like the left-hand side, right? Right? Right. Come on, stay with me here. Okay, so that leaves out predation. That suggests that it's actually a density effect, and if it's density and perhaps frequency dependent, then it's potentially an eco-evo interaction. Now, what do I have to show you in the way we've defined eco-interactions to make the case that these eco-evolutionary feedbacks drive the contemporary evolution in guppies? And we know that these changes occur very rapidly. Well, first of all, guppies must affect their community and ecosystem. That's the first part of that loop. The effects of guppies must create altered selective pressures that drive an adaptive change, and the adaptive changes in guppies must have qualitatively different effects on their community and ecosystem. We have to go around the whole loop 1.5 times. So, is this true? I'm going to summarize the results of a variety of experiments performed in artificial streams, or mesocosms, whole pool experiments, and some experiments inside pools where guppies are excluded by these electric fences on the left-hand side. You can see that it's green on the left, not so green on the right. Electric fence control. So I'll just summarize these results. First, do guppies affect their community and ecosystem? These tiny little fish actually have an impact. Actually, it's true. If you add guppies or increasing the density of guppies, it will reduce algal biomass. You saw that in the little picture. It will decrease invertebrate biomass, reduce killifish density, decrease leaf decomposition rates, decrease area-specific gross primary productivity via changes in algal volume, and increase the nitrogen flux if the guppies are high predation guppies. It's hard to imagine that a little fish could have this much effect. Yet, the data show that they do. So let me jump to number 3. Why is he jumping to number 3? Because the talk is about number 2, and I have to save that for the end. Plus numbers 1 and 3 are, in a sense, the low-hanging fruit. These are experiments that can be done in rather short term, meaning a year or so, whereas the middle one takes many years. So, do adaptive changes in guppies have qualitatively different effects? Are there significant differences between low predation and high predation guppies and how they affect their ecosystems? Well, this is the effects of exchanging phenotypes. Low predation guppies are more herbivorous, less selective in what they forage on, and excrete more nitrogen than HP guppies. Compared to high predation guppies, low predation guppies have a net effect of reducing algal biomass, increasing invertebrate biomass, not decreasing as we saw generally in adding guppies, increasing leaf decomposition rates, decreasing area-specific gross primary productivity via a change in algal diversity, not algal volume, and decreasing the nitrogen flux at low density, increasing it at high density. So there's some fairly complicated differences between the two types of guppies and how they affect their ecosystems. That means the only thing left to complete this loop is this one. The effects of guppies must create altered selective pressures that drive an adaptive change. In other words, we have to show that they evolve in response to a change they make in the environment and complete this loop. Now we think, by the way, that the environment involved here is population density. That guppies, by growing and having high density, affect their own selective pressures. We have this notion from some mesocosm experiments, and I'm going to zip past that and get to the time course of rapid evolution because the big bong will go off soon. One can do this the hard way by transplanting high predation guppy stock upstream into reaches without guppies, but with rivulus, and these are what the barrier waterfalls can look like. The idea being to do experimental evolution in the field. This is where we did it in 4 streams in a paired design, 2 introduced in 2008, 2 in 2009. The data are monthly mark recapture statistics where we monitor density and the phenotypes, and then F2 common garden results to get at the genetic component. So what we're going to do is try to match up the genetic component of these phenotypes, change over time, against the density of their grandparents 2 generations before. So the first thing we see is that there was much higher survival in these introduction streams than we typically see. The solid line is high predation, the light line is low predation. The dots are above the lines for the most part in our 4 streams. This produced very high densities and rapid population increase, as you see here. The rapid population increase also resulted in the same kinds of effects in our focal streams that we saw in other experiments. The same kinds of effects of excluding low predation guppies and high predation guppies when we looked at the resident guppies downstream and the introduced guppies upstream. We watched, this is from Fraser and Lamphere, killifish density drop dramatically, so they're affecting their environment. The densities are getting very high, and as the densities get high, the somatic growth rate drops. They slow their growth because they become food limited. And then, if you actually look over time, this is from the common garden experiments. This is the difference between the age and size and maturity, age in the top panel, size in the bottom, and the controls. They're getting bigger, they're getting bigger, they're getting bigger. And here's the relationship with density. So I claim to have told you this was an important question. The guppies are useful, and we've seen that density predicts their evolution in a way that makes it work. So thank you for coming. I'm out.